Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Went, and I'm the founder of Everything Epigenetics. Today, my guest is Dr. Charles Breeze. We touch on a lot of things regarding epigenetics, of course, but there are two main focuses of our discussion today. Number one, we describe the lack of diversity in these epigenome-wide association studies, or these EWAS studies. We talk about those current challenges and how we can have solution to those challenges and move toward a more representative data population. The second thing we discuss is several developed bioinformatics software tools that he has created called eFORGE and FORGE2. These are for the analysis of epigenome-wide studies and genome-wide association studies as well. A little bit of background into my guest today. Dr. Charles Breeze joined the Occupational and Environmental Epidemiology branch in 2020 as a postdoctoral fellow. He received his PhD in computational epigenetics and genomics from University College of London, where he was an EU Marie Curie pre-doctoral fellow. And he's even worked for some other leading institutions, including the University of Oxford, the European Bioinformatics Institute, and the Altius Institute for Biomedical Sciences in Seattle, Washington. This is where he actually led an integrative analysis and generation of reference mouse and code data. Dr. Breeze has developed several bioinformatics software tools, which include eFORGE and FORGE2, which I mentioned will be part of our second half of the discussion we have today. And now for my guest, Dr. Charles Breeze. I hope you enjoy. Welcome to the Everything Epigenetics podcast, Dr. Breeze. Thank you so much for being here today.
Thank you for having me.
I'm excited to just hop right into it. And really, this is the first time you and I have ever really spoken over the phone. I'm very familiar with your research, but would love to just hear a little bit more about your journey, how you became really involved with epigenetics. Tell me everything about the epigenetics, and just kind of your upbringing as well.
Definitely. So about the genomics or epigenetics, my first contact with genomics was really when I was 16. So I was basically, a student in high school, and my father had just gone off on a trip to the north of England, where he had been discussing ancient history with some archaeologists. And one of them lent my father a book on genomics, which was called Genome by Matthew Ridley, who's he's a very eminent science writer. And I just fell in love with genomics at the age of 16. I read that book, absolutely loved it. It had all kinds of sort of different stories. Each chapter was a story about a specific gene, and each chapter was also a chromosome. So it had, the full set of chromosomes in the human genome, and it went through. It took one example of a gene for every chapter, and it focused, for example, on Huntington's disease, or on height, or on BMI, and it really focused on all these different, very human aspects, taking genes and individual genes as kind of like an anchoring point. And I absolutely loved it. And from then on, I knew I was going to do research, which would eventually lead to epigenetics, which is, in my view, very much part of genomics, but much more exciting because of the potential environmental aspect as well.
Yeah, I wish that's a beautiful story. First off, I wish I had one that started, that young. It seems like you really just ran with it. You knew you were interested in it. So I appreciate you sharing that. And then talk to me a little bit more about your PhD in that journey. I know you've received your PhD in computational epigenetics and genomics from University College of London. So can you tell me a little bit about that, and maybe why you chose that particular pathway?
Definitely. So I was doing internships before the PhD just to get a good feel of what research would be like. And I was doing this internship at Oxford University on microRNAs. So these very short RNAs that have many roles, especially in cancer, but also in other conditions. And I was just, at that point, I was getting really interested in epigenetics. But of course, some people consider, the whole RNA, microRNA world as part of like epigenetics. And some people focus more on, DNA methylation and, and histone modifications. So it's kind of counterintuitive, but I started from the microRNA kind of like research angle. And I was really interested in trying to understand how environmental exposures can really, eventually mediate all this genetic risk, right? Mm-hmm. So there's long been a debate of, the nature versus nurture, right? I mean, is it something that we're born with, or is it something that we kind of acquire over the lifetime? And the real answer, at least from Matthew Ridley's book, was this is nature via nurture. So we have genetic predispositions to some things, but then, in our lives, we eventually have a series of exposures that can lead to a potential outcome. And so it was this nature via nurture, trying to understand how this genetic aspect and this environmental aspect come together, that really led me to epigenetics, to try to understand the answer to this problem.
Yeah, I really like that, the nature via nurture. I don't think I've ever heard it be put that way. I'm definitely going to start using that now, because that makes sense. Hey, we have these, genetic variants or the sequence we get from mom and dad. How are we able to affect those through the environment, through these epigenomic changes? So going to start using that now instead of versus. I think that's to distinguish both of those in that way is very important. And that's great that you actually started out in the RNA, the microRNA modifications. I think when we think of epigenetics, some people usually just think of the DNA methylation or the histone, acetylation, but the RNA is still there and so very much important. Definitely.
And RNA modifications as well. Chemical markers on top of RNA is also a really cool field.
Mm-hmm, mm-hmm. Yeah. Absolutely. And something that definitely not my expertise. So would love to hear about that more. But first want to really hop into your more epigenomic papers that we're familiar with. You do have one, Dr. Breeze, that was published in 2021. This is actually titled Epigenome-Wide Association Study of Kidney Function Identifies Trans-Ethnic and Ethnic Specific Loci. What does that mean? Can you explain that to our listeners and what you did in that study?
Definitely. So the study the title is long and complicated, but the reality is quite simple. So African Americans are actually 3 times more likely than European Americans to have kidney failure. Mm-hmm. And Hispanics or Latinos are actually 1.3 times more likely to have kidney failure compared to European Americans. And so this is a huge disparity. This is one of the biggest health disparities out there. And so what we did with our study was to try to understand, using genome-wide DNA methylation, to try to really understand the potential environmental and, of course, this genetic interplay behind this huge health disparity. And we wanted to really find target genes that we can really sort of follow up to try to eventually have better preclinical models, because without that, we can't really start addressing this disparity.
Yeah. And what was can you talk a little bit more about exactly what you identified there? I'm assuming you looked at, very large populations, some African Americans, the Hispanic and Latino population compared to the Caucasian population. So were you just looking, using the Epigenome-Wide Association study to look at those differences, or, how many positions did you find, and what did that really, I would say, all entail?
Definitely. So this is a 2-tier design study. So it has a large discovery section. There's a large discovery population, but there's also a large replication effort to really follow up and identify robust findings. And so in our initial first study, we analyzed 5,428 individuals. So over 1,000 African Americans, over 1,000 European Americans, and around 800 Hispanic Latinos in an initial scan to really identify, in the first pass, what DNA methylation positions are associated with kidney function. And we identified 93 CPGs that are actually significantly affected, or they're actually significantly associated with kidney function. So this is the discovery phase. And so we conducted a replication study with over 8,000 individuals. So an even larger replication study to really identify reliable results, essentially that could overlap with the initial study. So as we have 2 studies which are pointing to these sites. And we found that around 13 CPGs across different ethnic groups actually replicated well. And so these are key CPGs for follow-up to really try to understand the fundamental biological association of those CPGs with kidney function. Mm-hmm. So this 2-tier study is actually the design approach that we took.
Yeah, no, I think that's a great design approach as well. And I'm hoping my listeners know by now why replication is so important in these types of studies. I had a really great episode with Karen Sugden on that from Duke, really talking about this. Hey, if we find this association, we now need to validate it and replicate it in several studies. So you have these 13 CPGs in the second part of the study that were replicated really well. And you're then saying that those 13 CPGs are responsible for the difference that we see in those different populations for the kidney disease?
So those 13 CPGs are kind of like a marker or a strong marker for important genes and pathways that are associated with those differences. Okay. Of course, I want to caution that, this is the largest EWAS, epigenome-wide association study of kidney function to date, mm-hmm. but we're still early days in epigenetics, and we mm-hmm. still need to increase sample size to really reach levels at which we can actually explain a large fraction of the variability. Yeah. So these 13 CPGs don't mean that we understand the entirety of the difference, but they're a very good marker for pathways which are underlying that difference.
Absolutely. Yeah, that's great. And I said kidney disease, I meant kidney function. So I'm sure there's going to be, this is really groundbreaking research. If anyone's wanting to look into the function of kidneys, of course, or any type of disease relationship to those kidneys, these are the 13 CPGs that we would go to probably for that area of interest. Is that a correct statement? Perfect.
Yeah.
I want to make this easy for our listeners to understand and see, the value of this research that you've performed. Now, Dr. Breeze, correct me if I'm wrong. However, I believe, after talking from a couple of people from your team, in the process of this study that we're talking about, when you performed it, you really realized that the current analytical tools using the EWAS, the Epigenome-wide association studies, are based on European data, which of course is not going to be it's not going to be an all-encompassing sample size that for analyzing data from Latin and African populations. So how did you come to that realization, and what can we do about it?
Right. So this is a core issue in epigenetics right now. This lack of diversity in a lot of epigenome reference data. So this is not just for EWAS, but also for GWAS, which are the genome-wide association studies, which are some sort of the genetic equivalent of an EWAS. But whenever we do an EWAS or a GWAS, we always have this post-EWAS or post-GWAS section where we try to understand the fundamental biological pathways. Mm-hmm. And so we use a lot of reference epigenomic data to try to essentially map these things to genes, to regulatory elements, and to underlying pathways. And what we found was, we did this study across African Americans and European Americans and Hispanics, Latinos. And when we were trying to do the post-EWAS section, we really found that a lot of the reference mapping data didn't align very well with the African American or Hispanic Latino data. So we're doing these pathway analyses and these integrative analyses, and we're finding that the European data maps really well with the kidney references. But outside of Europeans, it doesn't really work very well. Yeah. And so this is a huge obstacle, because it's not just an EWAS problem, it's also going to be a problem for GWAS interpretation and actually for the whole of precision medicine. So epigenomic references are very important, and getting those right is going to be a crucial step forward.
Yeah, so I'm curious, why is this? I feel like you hear about these issues all the time, right? The diversity and the sample populations, and even things like sample size, and we can get into some of the critiques of study designs here. But why is this really the first time we're saying, okay, this is an issue, let's do something about it? This led to a lot of your recent work that was published last year in 2020. You reported a lack of diversity in the EWAS, which we've been chatting about, and then DNA methylation data. So, why are we just now hearing about this? I think that paper that you wrote is very, very important.
Definitely. So there's been somewhat of a change of perspective in recent years. So a couple of years ago, there was a focus on trying to make GWAS more diverse. People realized that GWAS data was not very diverse. And what we really found last year was essentially the same applies to a lot of other precision medicine, especially EWAS and epigenomics. The individuals and samples that are taken are overwhelmingly European American. And so this isn't really including everyone. And I mean, research only really works for all of us if it includes all of us. And that's not what's happening right now.
Yeah. Are there any other challenges you would want to discuss for the EWAS and DNA methylation research in diverse populations? Maybe some, other solutions as well? Definitely.
So one of the key issues that we found is that often for example, there are specific genes that are known to be very well associated with increased kidney disease, for example, in certain populations. So, for example, there is this gene, APOL1, and it's associated with increased risk of kidney disease in African Americans. And so, while this gene is definitely important to study, there is relatively little sort of epigenomic focus on technologies to really study that region of the genome. And so we really need targeted approaches. We need approaches that are aware of the diversity that exists in epigenomics and in human populations, and technologies that really embrace that. And so we think that obviously things that have been developed to date have been useful so far, but we really want new technologies, and especially technologies that are targeted towards improving health and detecting associations in regions that are critical for health conditions. For example, APOL1, and there's a number of other genes as well that you can list that would be critical to understand diversity and disparities in health.
Yeah, absolutely. I know there's, a lot of work being done kind of in the imprintome region area and epigenetics too, with, in particular, African Americans and, our Europeans, because there's going to be a difference for Alzheimer's disease, a very, I would say, large discrepancy. So as we're able to understand, I think, those differences across populations according to disease type, we'll be able to give a little bit more information. Now, Dr. Breeze, you also proposed several solutions for this lack of diversity. And I think these are, of course, some common solutions that all sound great. We just need to put them into practice. But these include things like getting people involved, getting people, being able to volunteer for different studies, more data generation, which I think is exactly what you were just hinting on. And then obviously some cost-effective approaches such as locus-specific analysis and ancestry variable region analysis, again, which you kind of touched on as well. So do you truly believe that we can kind of move towards this more representation based, studies or do you still think that's a little bit far away? How much time are we going to take to put this into application and move toward a more representative data sample?
That's a good question. So we're definitely in the first steps of addressing some of that. There are some smaller studies which have been done so far trying to understand diversity even in epigenomics. So, for example, TopMed, which is this extraordinarily large study focusing on precision medicine, has a subset of individuals and subset of participants which have been evaluated for DNA methylation. And so there's some really exciting work coming out of there. I think we definitely need studies that are targeted at addressing we need efforts which are focused on addressing this disparity. So focus on, for example, increasing Hispanic samples and increasing epigenomic analyses in Hispanics. So targeted approaches in technology, but also targeted approaches in terms of studies. We need studies that really go after trying to solve the gap. Just doing more studies, larger studies across everybody sounds great, but what we really need is also work in specific populations, right? So work in Hispanics, work in African Americans that's really like focused on increasing the numbers.
Yeah, I'm happy to have this conversation. I think it's something, hopefully people hear us talking and say, I don't think anyone would ever disagree, but I think that, getting the word out there, actually talking about this will make people get more involved as well. I'm really happy to say that with this podcast that I've been doing now, basically for a year, I've talked to people with really amazing backgrounds who are doing the studies themselves. So one comes to mind, Dr. Yanitza. She's actually out of Yale, and she's Hispanic, and she does, all of these really nice epigenetic DNA methylation Hispanic-based studies. So focusing on one different group and then being able to see those differences and compare them maybe from a larger population standpoint in the future will only lead to, helping this cause that in terms of needing more diverse sample types. So I think it's coming. The future is bright. I am excited about it. And then anything else you want to add there, Dr. Breeze, regarding epigenetic work and diversity? We're getting ready to move on to our second subject, which I'm really excited to talk to you about too. But any last comments or anything we can do better just as a community with, making these cohorts or EWAS or GWAS more diverse?
Definitely. I think also sort of essentially defending the value of epigenetics research, especially because health disparities aren't just a genetics problem. We've talked about APOL1, but they're also an environment problem. And so, for example, there's huge issues, for example, in water quality and in other environmental exposures and conditions, even within the United States between these different populations. And so it would be extremely valuable, I think, to sort of become more aware that epigenomics is not just a proxy for genetics, but it's also a proxy for some environmental exposures. And a real way to understand that better is going to be by doing more epigenetics and by doing more work in that area.
Yeah. No, thank you. That was beautifully put. I'm glad I asked if you had any other comments. That's great. I think I just got, I'm brand new to this world of epigenetics. I don't know what I don't know. We don't know what we don't know. I'm learning as we go on. This is a big part of why I'm having this podcast, is to talk to experts just like yourself in this amazing field that's growing very, very quickly. So, of course, I'm really more familiar with epigenetics as it relates to aging and those biological clocks and longevity. But, of course, this is very, very important work. And, I've just learned so much. And again, really have only have known the world of epigenetics since 3 years. And, the research just continues to expand. So I'm excited to see.
Definitely. And I also want to add, why DNA methylation, right? So everybody who talks about epigenetics, there's all these different marks. We've even talked about microRNAs, and we've talked about histone modifications, but DNA methylation it's not a surprise that DNA methylation has emerged as one of the major marks in epigenetics. And one of the reasons for this is that it changes with environmental exposures, but is relatively also has a certain stability throughout the lifetime. And so, for example, as you've discussed in previous podcasts, it's an extremely good marker for aging. But it's also a very good marker for smoking and other very long-term exposures. I can recall the case of one participant in a study who essentially we stopped smoking several years before, and subsequently when we evaluated the participant a few years later, this was 5 years later, still had some signatures of the smoking mark. So even though he had stopped smoking, there was actually still a mark there, an epigenetic mark, right, of that exposure. And so a little bit like a sort of a record of all lifetime exposures, DNA methylation is just an excellent choice.
I'm so glad you said that. I'm so glad you said that because I will say this. I'm not going to name names, but some of our researchers actually from Cornell that I work with very closely through my work at True Diagnostic, they actually came to visit our facility. It was really nice. We love, showing off our lab here in Lexington, Kentucky. But we were basically talking to them and they said, you know what? A lot of our friends, they're disease experts, but a lot of their, colleagues and friends have always been really riding on the fact that the transcriptome looked to be the best when studying infectious disease. They've actually almost shifted entirely to epigenomics and to DNA methylation as that modification. And I just think, epigenomics, the DNA methylation, it really does seem to be this underlying almost like kind of gold mine for understanding, the how, and not only the how, but maybe the why eventually. We definitely are starting to get the correlation, causation. We'll see what we can understand there. So I really do. I think DNA methylation acts as a great proxy to be able to start and to study. I think what you said, Dr. Breeze, is really great because, again, when people think of epigenetics, they think of change and they think, oh, this changes really much. But again, DNA methylation is still stable enough to have and hold on to that signature across the entire lifetime where we're able to see what we've been exposed to. So there's a lot of value there, a lot of things that can be studied. All right, Dr. Breeze. Now, really excited to talk to you about a couple bioinformatics-based software tools that you've created and developed. And you're probably going to have to break this down a little bit more for us here. Now, do you pronounce it eFORGE and FORGE2?
Yeah, eFORGE. Yes.
eFORGE?
Exactly. Yeah.
Perfect. So this is, the analysis of epigenome-wide association studies, EWAS, and then those GWAS studies as well for that data. So I know you just expanded your FORGE database. So, can you explain what this is? And I'm definitely going to be sending this episode. I sent your work even to our bioinformaticists at True Diagnostic because I just think this could be extremely helpful to people listening. Of course, our researchers too.
Definitely. So the names, the pronunciation is correct. eFORGE was actually named by the scientist who did the first human methylome. That's Stefan Beck. So it really goes back to a lot of DNA methylation work. But essentially, what these methods are trying to do, they're trying to essentially create robust, comprehensive, and unbiased frameworks for post-EWAS and post-UAS analyses. And so often we do an EWAS, an epigenome-wide association study, and we come up with a list of genes, well, specifically a list of CPGs in the case of an EWAS. And essentially, once we've done that study, we have this list, but we want to take that into the next step, which is to identify fundamental pathways, biological pathways which are associated, and try to get some biological insights into, for example, kidney function or the specific condition that we're studying. And so often what's done in the post-EWAS analysis world is to try to do enrichment analyses. And so people have all kinds of pipelines and different types of software to evaluate that. And sometimes this is a bit ad hoc. So somebody just runs a couple of R scripts and comes up with some enrichment results. And what we wanted to do was really create a sort of unified and robust framework for conducting these enrichment analyses. It's a little bit like herding cats. There's all these different methods, and we wanted to create some unified approach. So basically, that's eFORGE, and it's basically just trying to really come into the robust, trying to come into a robust framework for something that's always been so, chaotic.
Yeah. Now, no, that definitely makes sense. Can you just define what the enrichment analyses means, or what that all pertains?
Definitely. So when you do a study, you will oftentimes want to know what regulatory elements, what genes, what enhancers those key sites, those CPGs are coming up in. And so, for example, you will do an EWAS on kidney function, and you will find all these CPGs. Some of them are intergenic, right? So we don't know what they do. Some of them may be in a promoter of a gene, so you have a slightly better idea. But to really understand this at a systems level, we need to take an approach that's called integrative epigenomics. So taking epigenome reference data and trying to integrate that with the results from your study to try to see what key enhancer components, what key tissue components, what cell types are really associated with your results. So, for example, in the case of a kidney function study, you can do integrative epigenomics to try to find out, is it the nephron that's really coming up? What pathway inside the nephron in the kidney is really the important pathway? And so this is all an effort to try to link this to fundamental biology and try to understand the fundamental biology behind your epigenomic results. And so that's what eFORGE has really been all about. It's really been about doing integration and doing it in a robust way. So eFORGE always analyzes the whole set of tissues in the human body. And so sometimes, when people do post-EWAS analyses, they only analyze, some T cells and, and some B cells, and they come up with a result. But how does that look across the entire set of cell types? That's what we want to know. So it's all about robust and comprehensive frameworks to really come up with reproducible results. Yeah,
that's great. Again, I'm if I could go back to school, I would definitely do some bioinformatics works or like some computer science. I'm more, general biology based and genetics. So I guess to understand this, would this be correct, Dr. Breeze? You have all of this data after an EWAS study. There's a lot of different normalization pathways and pipelines, and it's messy. It's not necessarily regulated. Here comes eFORGE. You're putting in all of that data, and then that's going to tell you kind of, again, it's going to normalize, I guess, a lot of that information, and then maybe even tell you, more of like the mechanism of action. How can we make this applicable and take it out to our true biology and relate it to our fundamental understanding. Is that correct, maybe?
Definitely. And I would also add that one of the focuses of eFORGE is so essentially what we're trying to do here is not just find the underlying pathways, but also correct for all these different sources of bias. So eFORGE under the hood has different methods for bias correction, just to make sure this is all done properly and you're not getting spurious results. So so definitely that's a good summary.
Well, that's great. Is this open source for anyone to use? Are there any costs involved?
100%
open source.
Open source, and all of the versions are publicly listed on multiple databases, GitHub, Zenodo. We have all kinds of backups because we want to keep this open source for the community. Yeah.
How'd you create this? I imagine it took a very long time.
It did. It was interesting because actually my background, I actually started in the wet lab. And so when I was doing microRNA work at Oxford, I was 100% wet lab. And it was interesting. I started getting more into computational biology and really sort of learning more about programming over the years. And essentially during the PhD, that's when I really worked on creating eFORGE. And yes, it did take a long time, but it was definitely worth it.
Yeah. Well, I think it's a beautiful tool and what I'm going to see if we can play with a little bit. So just to confirm as well, the eFORGE is to better understand the EWAS, and then the FORGE2 is to better understand the GWAS. Is that correct?
Definitely. And of course, EWAS are very different from GWAS. So FORGE2 is actually really different from eFORGE, like in many of its underlying assumptions. But the key result is the key objective is to unify, to create a unified framework to really do these analyses properly, and definitely so we can translate the value from all the value that's coming from the study to eventually translate that into preclinical models and into work that can eventually lead to essentially results for people who are taxpayers who are funding all of this work.
Perfect. And you kind of hinted at this as well, Dr. Breeze, when you were talking about the EWAS study. This is a reoccurring theme on my podcast. So listeners, if you're tired of hearing about it, I'm sorry, but this is very, very important. What about the cell type specificity? You said, if you have an EWAS study and you're trying to, again, find the pathway, what's happening in the body, they may be only looking at one certain cell type. So you can even redefine it for our listeners, maybe who are just now, listening to the podcast and are new, but what is cell type specificity and why does it matter when we're using, eFORGE or EWA studies?
Definitely. So cell type specificity matters for almost all areas of epigenetics. You've previously mentioned epigenetics of aging. So in epigenetics of aging, as you know, you have extrinsic epigenetic age acceleration and intrinsic epigenetic age acceleration. And that extrinsic or intrinsic is whether the changes happen within the cell or because of changes in cell composition. And so this is critical to understand aging. It's also critical to understand every UAS that's ever been done is critically dependent on cell composition. So as you know, this was a bit of a scandal at the very beginning of EWAS, but very first EWAS were trying to find results in blood. And of course, blood has B cells, it has T cells, there's many different cell types. And, what people found, essentially Hausman and several other researchers, what they found was that you have to really correct for these cell type proportions because they can drive your entire study results. And thankfully, that was found very early on. And subsequently, every single EWAS that's been done has adjusted for cell type proportions, or at least there's been a focus in the field to adjust for cell type proportions to really make sure that your results are driven by changes in DNA methylation that are happening, inside each cell. And so essentially, cell type specificity is crucial for EWAS, is crucial for aging, and it's really also crucial for understanding the fundamental biology. So when we do these post-EWAS analyses, we really want to know which cell type these enhancers are active in that are overlapping these sites from our study, and really trying to map at a cell type level so as we can translate this to precision medicine. So, for example, in the case of kidney function, which cell type in the kidney is the one that's really driving all of this change? And how can we know more about the pathways that are happening there? So trying to understand biology, I come from the wet lab, I really love biology, and trying to understand the fundamental take-home message, that's critical for me.
Yeah, no, I can definitely tell. Your passion is seeping through as you're talking, so I can definitely see it there. And that's always extremely nice to know your life work and everything, something you're really passionate about, and you feel very, very strongly for. So yeah, cell type specificity is absolutely crucial. And you made a good point. Good thing we found out now. Could you imagine if we released all of these studies and then decades later, everything we've ever known? I just couldn't imagine. So yes, that is, I think something I struggle with every day when I talk to, healthcare providers or researchers, and some people aren't even informed about this. And I think it's really important when we're looking at epigenetics, even more for the aging aspect, especially when people are starting to use some of these clocks for more of a commercial aspect. So, Dr. Breeze, you've actually brought up, epigenetics and how it relates to aging. Well, I've brought it up, I think, and have pushed it on you and asked you about it. But are you very familiar then with the epigenetics and aging? I just have to ask because the audience is, I'm sure, very interested in that as well. Or do you ever plan on doing any work in that space?
Definitely. So I've collaborated with Steve Horvath on several studies, and actually there is a specific version of eFORGE which was developed for one of his aging studies. So there's definitely a lot of focus on aging and its relationship with multiple human conditions. For example, aging and cancer is a huge field. So yeah, I've definitely worked on aging work. The fascinating thing, and one of the really open questions for aging is fundamentally, how does the epigenetic clock really work? And so Steve Horvath's clock, which, as you know, was developed, published in 2013, came out right around when I was doing my PhD. It's an extremely precise biomarker of aging, but nobody really understands at a fundamental level how it works. And so we actually teamed together to do more eFORGE analyses on the epigenetic clock. And what we found was there's some strong associations, for example, with the repressive polycomb complex. But that needs to be followed up. So there needs to be more work done to understand the fundamental biology of aging and what's making the epigenetic clock tick. Well, we don't know that, but we're trying to find out more.
Yeah. I'm going to, when we stop talking online, I'm going to talk to you offline about some opportunities that I just thought of. But yeah, what would be interesting, Dr. Breeze, is Dr. Morgan Levine came out with a really nice paper where she creates these like CPG, I think she calls them modules, where she compares all of the really famous, and I would say that, the best of the best, the most precise, sensitive, accurate epigenetic clocks that have been created to date. And then she creates the overlap of these modules in terms of the CPGs. So it would be really, I think, interesting if you were to take those CPGs that overlapped in specific clocks and actually ran them through your eFORGE to see, all right, where are they overlapping in terms of, again, the functionality, right? The mechanism of action there. So I think that's just one of the many ways we can start to put your really nice bioinformatics software tools to use.
Definitely.
Yeah, go ahead.
No, I was also going to say, of course, for aging, people have also tried to do GWAS to try to understand the clock. So trying to go back to genomics and trying to understand DNA methylation changes. And there's definitely also applicability for FORGE2 there. So, I mean, these methods are obviously going to be very useful going forward. And and that's definitely a good idea that you mentioned there.
Yeah, absolutely. Excited for what's to come. Now, I should have asked this at the beginning of the call, I should say, but why kidneys? Why? How did you get started there?
Good point. So one of the major areas where the environment really ends up affecting overall health is through, for example, through water. So we all know that there's substantial exposures to lead and to other toxins. And some of these, especially, for example, for PFAS, which is this group of, organic compounds which are found to be in the water in certain parts of the United States, all of these different exposures, oftentimes they reach people through water. And it's the kidneys that really filter not just the blood, but they act as a filter for a lot of toxins. And so this was really one of the most exciting areas, I think, to do epigenetics in. So obviously there's other organs which are crucial. The liver is crucial for exposure to toxins. And there's a number of other approaches that one can take. But to understand kidney health, that's tightly linked to health disparities. As I said before, there is a substantially higher likelihood of kidney failure outside of European Americans. So to try to understand health disparities, we really have to focus on the kidney.
Yeah, no, I appreciate that insight there. That definitely makes sense and makes for a good, organ to study. And, hopefully we can figure out why those health disparities are happening through that kidney function and filtration system, so to speak. So we're getting toward the end here, Dr. Breeze. Just a couple questions left for you that I want to pick your brain about. What's next? What are you most excited about? What are you working on? Is it some of these more diverse, inclusion type of population-based studies? Are you focusing on a certain population? Are you working more on any of these eFORGE or FORGE2? What's next for you?
Definitely. So I'm glad you mentioned the kidneys, because our next big study is going to be on kidney cancer, to try to really understand how diversity affects kidney cancer and DNA methylation. So that's going to be our next big effort. I'm really excited to do more work on that. And we're trying to get that done as soon as possible, because all these epigenetics results can't wait. And the next big project, apart from the focus on kidney cancer, of course, what we're trying to do is also expand the pathway approaches. So you can do pathway analyses, taking data from eFORGE and feeding that into pathway analyses. But what we really want is to integrate the epigenetics and the pathway software together very closely, so as we can really map pathways in individual cell types that are essentially linked to study results. So we're trying to bring the wet lab mentality and computational biology really close together, so as we can get more information on the fundamental biology, which I think is the most exciting part of any study.
Yeah. I mean, I couldn't agree more with that. Well, I'm really excited for that study to understand, again, the kidney functionality and the cancer and how that all ties back into the genomic profile, the epigenomic profile, and then how we can hopefully make that an application for users too. So, Dr. Breeze, one last question on the end of the podcast. I usually ask everyone, all of my guests, if you could be any animal in the world, what would you be and why?
That's a very good question.
It's not on the agenda that I send beforehand.
So that's pretty funny. So my wife, she has these rabbits. So I'd probably want to be one of them, actually, because they get the very best life. They get treats. They get all kinds of snacks. I mean, sometimes I think my quality of life is actually kind of like on par. So like if I was going to keep, my quality of life going, I'd probably be one of those rabbits. Yeah.
Oh, well, very good. How many rabbits do you have? You said just 2?
There's 2, and they're actually resting. So yeah, that's what we're up to. Yeah.
Well, good. Sounds like a really good life. Well, Dr. Breeze, we've come to the end of this amazing podcast interview. For any listeners who want to connect with you, any bioinformaticists or research who want to get access to the eFORGE, where can they find you? Where can they get these tools?
Definitely. So I'm always on Twitter, and I'm also really involved on GitHub. So if they can reach out on either one of these social media or computational web pages. I'm also definitely so the latest release of FORGE-DB, by the way, that's all public. So all of these web pages are linked, and I'm also available by email. So I would love to chat about that.
Perfect. And I'll make sure when I publish this on my website that I put all of the links. That way people can, go to your profile and you can get that exposure and people can contact you as well. So thank you, everyone, for joining us at the Everything Epigenetics podcast. And remember, you have control over your epigenetics. So tune in next time to learn more. Thanks, Dr. Breeze.
Thank you very much.